we are finally diving into what I think is the beginning of our true look into biology, and that's diving into the molecular, how our cells and our bodies operate on a chemical level. So we're going to talk about biomolecules, the molecules that make up life, and enzymes. Enzymes are quite fascinating and one of my favorite things to teach about. So first, starting with this idea of our biomolecules. Here's all of the important key terms for you to know. We have to start with going over one of our main themes in biology, and that is that whatever the structure of a body part, a chemical, an organism, whatever its structure, that will give you a hint as to what its function is. So structure informs the function. And this does go all the way down to a chemical level, and that's what we're going to get into a bit today. Now, really, this goes all the way back to physics, because the laws of physics are what determine why our chemicals interact with each other the way they do, why our molecules and our compounds take the shapes that they do, and from there, that informs how they behave with each other and how they behave in a broader landscape as well. So once we have this understanding of the structure, then we can start to piece together how it works. So with chemical shapes, for example, we've been talking about water. And if we know that something's polar versus nonpolar, we know that it can interact with water if it's polar and it cannot if it's nonpolar. So there's one hint already about how a chemical might behave based on just its polarity alone. So we have to start with this idea to talk about the biomolecules of how they're built. So biomolecules are molecules that are very large. We often call them macromolecules, large molecules. And they are what we call polymers. Break down that word, it means poly means many, mer means unit. So polymer, that's many units. So all of our large biomolecules are these polymers that have a repeated basic chemical structure. That repeated structure is called a monomer, a single unit. When these monomers are joined together chemically, we call that polymerization, making the polymer. And the bonds between these monomers are almost always going to be covalent bonds. So we can see here, there's a group of monomers. The monomer is that little green pill looking thing. And the act of polymerization makes those bonds between the different monomers. And water, once again, is the key to all of these polymerization reactions. So there's two major types of polymerization reactions, and that is dehydration synthesis and hydrolysis. One is forming, one is breaking. So with dehydration synthesis, it's exactly what it sounds like. Synthesis is we're making something, that's what to synthesize is, and dehydration is by removing water. So we are making something by removing a water molecule from the compound. So we can see that in this first graphic up at the top. These two separate chemicals have hydrogen and a hydroxyl group in their chemical structure, so we can form a bond between them that you can see right here if we remove that hydrogen and hydroxyl group and form a water molecule. So this is forming a polymer, forming a bond between two monomers by removing a water molecule. Hydrolysis is the exact opposite. It is taking one molecule and we're going to break that bond by adding in water. So we're going to end up with those two chemicals and the water split between them again. So hydrolysis, that literally means breaking using water. So lysis means to break or to burst, and hydro means water. So that gives you a hint of what's happening in that chemical reaction. And all life is made up of 4 of these large polymers. We call them lipids, nucleic acids, carbohydrates, and proteins. And we're going to dive into each of those right now. So we're going to start with proteins because they are going to be the one that requires the deepest level of understanding, and they have the most moving parts, I guess we could say, and they're relevant to what we're going to talk about later, which are enzymes. So proteins are the most numerous and the most versatile biomolecule in the cell. That means they have the highest number and they can do the most things in our cells. They are polymers that are made up of a monomer called the amino acid. So the amino acid is the monomer. We bond them together and we get the polymer that we just call a protein. And they're joined together by a bond called a peptide bond. So proteins make peptide bonds to secure their amino acids together. So you'll have like an amino acid right here, connected to another one, and those bonds are the peptide bonds, and they're a form of covalent bond. Now, proteins have very specific structures that we're going to talk about in a little more detail in a second. But they form enzymes, they form proteins that make up our membranes, they form things like insulin, toxins, hormones, our muscle fibers. So not only do proteins make up the structure of our bodies, but they make up chemicals that actually function in our bodies too, meaning they do a job in they play a role in our cells and in our bodies. So, as I said, the monomer, the single unit, that little green tic tac, is the amino acid, and they're joined together by a special cell structure called a ribosome, and they form a covalent bond that we call the peptide bond. So you can see here, this is one amino acid, and this one here is the second. And once again, there's that water molecule that we'll be able to break apart or pull out of it. Dehydration synthesis is going to happen. We're going to bond the carbon and the nitrogen together as a result, and that water molecule is going to be a waste product. And we end up with our peptide bond in between, linking the two amino acids together. And once again, structure informing function starts all the way at the very beginning at the monomer stage for our proteins. The reason why proteins have the immense variability in their structure and their function all comes down to the structure of the amino acid. So amino acids have a basic structure, and then they have a part of them that can go a little wild, we can say. So there's always a hydrogen bonded to a central carbon. Then there's going to be an amino group. That's where it gets its name, amino acid. So there's always a hydrogen, always an amino group, and then there's always a carboxyl group. This is where we get that acid term from. So the last chemical that can bond to that central carbon, we labeled it as R here. There is no R on the periodic table, and that's why we use this for a reason, because this is going to be a variable group. The R can be any type of chemical really that wants to be there, and we call those functional groups. They're groups of chemicals that perform specific functions. And these are going to differ from amino acid to amino acid. As a result, we name amino acids based on what they have in their variable R group. So these functional groups all have a specific chemical structure that differs from each other, and as a result, because their structure is different, they are going to function differently. So every amino acid behaves slightly differently than every other amino acid does. This is going to be responsible for giving the protein its overall shape, because these amino acids will then interact with each other in the polymer. It's going to allow them to interact with other chemicals, maybe proteins, maybe fats, maybe carbohydrates. And in all, there are 500 variants of amino acids. despite having such a high number of variations in nature, only 20 of them make up the living things that you see all around you. So even though there's 500 for us to choose from, every single living thing uses the same 20 to make all of its proteins. So kind of important that we understand at least these 20. So here's some examples of functional groups. They can be something non-polar, they can be polar, they can have rings, they can have double and triple bonds that makes them very strong, they can have acid behaviors, they can have base behaviors. It all just depends on what you have in that spot. And you can see all the different name groups of the different chemicals that can fit in that R group. And here are the 20 amino acids that are found in all living things. You do not have to memorize these. We will talk about them again later when we talk about how we actually take our DNA and use that code to make our proteins. But for now, you just have to know there are 20 and they make up all living things. And you can see the variation in their structures just from these images here. So we're moving away now from the amino acid structure and we're pulling out a little bit wider. We're looking at the overall structure of the protein itself. And there's 4 levels to this that we're going to talk about. And these are all going to have a key, other way around. The amino acid structure, those functional groups, are going to be a key player in how these 4 levels of structure take place. So first we have the primary structure. You can see that one right here up at the top. That is the order of the amino acids in the protein. So there's 20 of them total. Which one comes first? Which one comes second? Which one comes third? That is your primary structure. And so it kind of, when we depict it as like a necklace with a bunch of beads on it. From here is where we start to see those interactions between the functional groups. So some of them will bond to each other, some of them will repel each other, some of them will be neutral in all of this. And as a result, we get our secondary structure. This amino acid chain will start to fold into what we call helices. They're kind of like corkscrews, or pleated sheets. Those look like when we were kids and we would fold paper a bunch of times and make a fan. That's the same idea. So these interactions between the R groups, between the functional groups, create these alpha helices and these pleated sheets, beta pleated sheets within the protein itself. So within this long chain of amino acids, some areas are going to form these helixes, some areas are going to take on these pleated structures. So you can have many different helices on the same protein. You can have many different beta pleated sheets. You can have one and 10 of the other. It just depends on how these amino acids interact with each other. Then we're moving on to our tertiary structure. This is where all of these pleated sheets and alpha helices and other areas of the amino acid, or I'm sorry, of the protein start to interact with each other on a bigger level. So you will see right here, for example, there's a pleated sheet. Here's an alpha helix, and they start to interact with each other on a large scale level so that the entire primary structure has been folded over once. Now it's being folded over on itself again. And this is where the protein starts to become functional. So prior to this, there are very few instances of primary and secondary structure leading to a working protein. You have to get to this tertiary structure for 99.999% of proteins to work. Of course, there's always exceptions to every rule. That's going to just be something I keep saying throughout the semester. We are talking about the vast majority, the usual thing, not the exception. So here we start to form that functional active protein. A lot of proteins stop at this point, and they don't necessarily form the next level of structure. Once they become functional, they're good to go. However, some proteins can form what we call this quaternary structure, and this is where the light-colored blue protein interacts with the dark blue colored protein. So you have two different proteins interacting with each other. It can be two, it can be more. It just depends on what the proteins are and what the overall structure is. So this is where proteins interact with other proteins to make basically metaproteins. Now, obviously, if we're talking about proteins, the loss of their structure is going to be very important and very devastating to the protein itself. So if we get to tertiary structure, that's where the protein becomes active. Well, if something causes us to lose tertiary structure, we can assume the protein can no longer function. It won't be active anymore. So as a result, proteins have to work under very specific environmental conditions because all of these levels of structure are due to chemicals interacting. If you tend to add a little bit more of a positive chemical to the environment, it can rearrange how all of that protein functions because all of the structural interactions will change. Maybe all of the positive interactions get overtaken with this interaction with a positive chemical that you introduced into the environment, so the protein itself unravels. Maybe you added too much acid or too much base, or it got too hot or too cold, and the actual movement of those chemicals, because remember, all chemicals are buzzing around and moving around all the time. It's considered, just part of life. If you get to absolute zero and all molecule movement ceases, that's not a good thing for life. So as these chemicals are constantly vibrating and moving around, if you up the temperature and they start vibrating more, you can end up causing some associations to break apart, and that'll change the structure of the protein. So we call all of this denaturation. We denature the protein. And this is where the protein loses their function because they lose their shape. So this can come about, like I said, by heat, by cold, by adding too much acid, by adding too many electrolytes or positive or negative charges, anything like that. An example that you are very, very familiar with is cooking eggs. Eggs are protein and fat. And the white of the egg is pretty much entirely protein. It's a protein called albumin. And when you crack open that egg, it's in its working form. It looks like a clear liquid, but then you heat it up and that protein unravels and it ends up forming an opaque, hard, white structure, and it can no longer go back to how it was before. That is key. Usually, once again, there's always exceptions, but for the most part, when you denature a protein, it is permanent. You can't put it back to where it was before unless you have a special chemical that can do that, and even then, not every single protein is capable of going back to its same shape as it was before. So this is really important, and this is why when you're doing things in like a chemistry lab or working with chemicals, you're always told to wear goggles, because some of these acids and bases, if they get splashed into your eyes, they can cause the proteins in your eyes to permanently denature, and it makes you blind. So moving on away from proteins, we're going to revisit them a bunch, especially when we talk about enzymes, but let's talk about our next biomolecule, our macromolecule, and that is carbohydrates. These are the energy provider for the cell. That's a good way for us to think about them. They are chemicals that the body can break down quickly, so we can use them as an energy source. There are simple sugars that are smaller, and then there are complex or larger sugars that actually can be used to build up the structure of things like plants or fungi or things like that. However, it depends on what the overall structure is. Once again, the structure is going to tell us what its job is. So we break them down into two basic groups. There's monosaccharides and polysaccharides. The monomer, or the basic unit, the building block, kind of like the Lego piece that you use to build your structure, is called the monosaccharide. So this is the monomer right here. And everything else is going to be built off of monomers that look very similar to this. There's a bunch of different types of monomers, just like there's a bunch of different types of amino acids. This one just happens to be glucose that I'm showing you, but there's lots of other types that can be used as the base here. When you put two of these monosaccharides together, you form a simple sugar called a disaccharide, like this one here. We call it sucrose. And then once we start adding more than two, we get these long repeated structures of these monosaccharides, and we call them polysaccharides. And the example here is starch. So breaking down these words, mono means one, di means two, and poly means many. And the term saccharide is basically, it's Greek or Latin, I can't remember which, but it basically means sugar. So we're talking about, do I have one sugar, two, or many? Kind of a little on the nose. Moving away from carbohydrates, because that's really all you need to understand about them. We'll talk about them once again a little bit later. We'll definitely get into them when we talk about photosynthesis and respiration. But let's move on for now. The next one are nucleic acids. These are probably the next most interesting after proteins, and we are going to deep dive into these a lot when we talk about genetics. These are the molecules that make up the code of life. They are the molecules that we call DNA and RNA. So their monomer, their building block, is called the nucleotide. Nucleotide for nucleic acid kind of goes together there. And they have many different polymer forms. The main two that we know of are DNA and RNA. You may have heard of ATP. It's the energy molecule for the cell. And there's other similar forms to ATP that also fall in this category of nucleic acids, but we're not really going to talk about them too much in this class. The main thing about these nucleic acids is that they make up our genetic information. They are very easy to copy and spread themselves. And because they have a specific way that we can create a code with them, that code then becomes our protein. So it's kind of like the blueprint or the recipe that the cell uses to make the proteins that then go out and make the structure and do all the jobs in the cell. And RNA acts like the messenger between the DNA and the protein. And we'll get into all of that much later down the road. So right here is a nucleotide. And the nucleotide has three main parts. It has a phosphate group. It has a sugar. We'll go into what sugar and why later on, because it does become a little important when we talk about DNA in our genetics unit. And then some kind of nitrogen base. This is just a chemical that has nitrogen in it. And we break these down and we then abbreviate them by the first letter in their name into A, T, C, and G. You've probably heard about that before. Then the order of those A's, T's, C's, and G's becomes the genetic code. The last biomolecule that we're going to discuss are the lipids. These are very large chemicals. They are most often nonpolar chemicals. We tend to mainly think of hydrocarbons, so chemicals that are just carbon and hydrogen repeated over and over and over again when we talk about lipids. Because they are very large and they have repeated structures with a lot of bonds in them, bonds contain energy. So this is how we store energy inside our cells. So we use them for that energy storage, and we can use them for protection as well from the elements, things like that. Some examples that we're familiar with: fats, oils, waxes, steroid hormones, things like that. So we have lots of different uses for them. We even can use them as chemical messengers within the cell. We're not really going to talk about too many of these. We're going to talk about the phospholipid that makes up our cell membrane. Otherwise, the rest of them just know that they store energy or they act as signal molecules like our hormones. These ones do not have a true monomer or polymer form because they can take so many different types of forms. As you can see with the steroids and our hormones, they take a lot of geometric patterns, whereas the phospholipids and triglycerides and fatty acids tend to just be carbon, hydrogen, repeated over and over again. So that is it for our biomolecules. We're now going to jump into enzymes, which are a lot of fun. Here are your key terms. So enzymes are proteins. This is something that a lot of students get mixed up on. Remember that every chemical that we're going to talk about, pretty much every chemical that we're going to talk about from here on out inside the body and inside the cell is going to fall into either, is it a carb, is it a lipid, is it a nucleic acid, or is it a protein? And enzymes are just a type of protein. They're a special class of protein that have a role in the cell. And that role is to speed up, or we use the word catalyze, chemical reactions. So they are going to make chemical reactions happen faster in the cell. They often have names that end in ASE, like catalase, and now I can't think of any others, lipase, polymerase, all of these different words. Without these enzymes, without enzymes, period, reactions would be so slow that no life would be able to exist on Earth. A lot of these reactions would happen on, levels of hundreds to thousands of years rather than the nanoseconds that we need them to within our bodies. So they are very, very essential to life. And looking at what we're about to talk about from a larger point of view, we're getting into energy here for a little bit. This is our first peek behind the energy lens. And we have to talk about metabolism if we're going to talk about energy. And metabolism is just all of the reactions happening in your body at one time. These can be catabolic or catabolism, the reactions that break molecules apart. So things like digestion, that's a catabolic reaction. We're taking our food and we're breaking it down into smaller pieces that make it easier to use and digest. The other side of metabolism is all the anabolic or anabolism reactions, and those are ones that bond molecules together to form larger ones. So building our proteins, building our carbs, making copies of our DNA, those are all examples of anabolic reactions in our body. Building muscle, that's why we think, anabolic steroids, you take them when you want to put on or build muscle. And so catabolism breaks down, anabolism builds up. And enzymes are key because for the most part, enzymes are going to be responsible for doing all of these reactions. So how do enzymes work? They function by what we call lowering the activation energy for a chemical reaction. We have to go back once again to basic physics and chemistry here. I've mentioned it a couple times now, but all chemicals are in constant motion. They're constantly bumping into each other, and they are vibrating and moving around with Brownian motion and just constantly in motion. So as a result, they will bump into each other. When they bump into each other, if they're at the right angles and with the right energy, they can form a chemical reaction. That's basically how this all works. None of this is a driven or purposeful pursuit. All chemical reactions just happen because the chemicals happen to be near each other, they happen to bump into each other, and it happened to bump into each other in just the right way at just the right time, with just the right energy. So something really important to understand before we get into this whole idea of enzymes. Covalent bonds in particular are also incredibly stable, so it takes a lot of energy to break them. They have to be in just the right particular structure and orientation with the, high amounts of energy input in order to break them. So when we talk about chemical reactions, in order to perform one, the molecules have to be first placed in the proper orientation, and then you have to have the energy to break the bonds and form the new ones. So how do enzymes play a role in all of this? So enzymes lower what's called the activation energy, the energy that is required to get a chemical reaction started. Basically, what this is that the enzymes will capture these chemicals as they randomly bump into the enzyme. It still is dealing with random chemical movement, but the enzyme will hold on to these chemicals as they bump into it and force the chemicals into the proper orientation with each other to force the reaction to occur. And as a result, because the enzyme is holding them there, it's making the reaction more likely. It's lowering that energy burden that would have otherwise been there to put the chemicals in the proper orientation and break those bonds. So if normally they have to get over such a high hurdle like this here, kind of think of it as like a really steep big hill, the enzyme will lower that hill and make it a little less steep for the reaction to begin. So there's still energy that is having to be inputted to create this chemical reaction, but it is far, far less than it would have been before. Now, when we're talking about an enzyme, we have to understand how its structure plays the role in creating these chemical reactions and speeding them up. So the substrate is the chemical that the enzyme is going to act on. It's the chemical that fits inside the enzyme and is either broken apart or two chemicals that come in and get forced together. And each enzyme has its own unique substrates. Once again, for the most part, every enzyme can only act on one type of molecule, one particular chemical, and either, bring them together or break them apart. So we call this like the lock and key fit. And the substrates are going to fit into the active site of the enzyme. It's just the proper shape of the enzyme where those chemicals can fit and then be forced into the correct orientation for that reaction to occur. And at the end, you are left with products, the results of the chemical reaction, either two molecules from the one larger one that you broke in half, or one molecule from two that you brought together. So as the substrate comes in, it is a lock and key fit still, in that only the correct substrate can enter into the active site of the enzyme, but there's one more step even beyond that, and that's what we call the induced fit model. What ends up happening is, as the substrate enters into the active site, the enzyme clamps down on that substrate even better, and the act of the substrate binding, it's not a true bond, but the act of the substrate coming in and interacting with the enzyme is what is going to force the enzyme to change its shape just a little bit, to force the substrate to change its shape to make the reaction happen. So everything happens for a reason when it comes to our bodies and the chemical reactions around them. And usually this interaction is due to hydrogen bonding. So the substrate and the active site are going to hydrogen bond with each other, cause the enzyme to shift its shape just a little bit, which causes the substrate to get forced into the right position so those bonds can then break easier or form easier. So all of this induced fit is just to better align the substrates for the reaction to occur. Another really key point with enzymes: they do not change as they catalyze their reactions. They're not used up. We call them recycled, because one enzyme can perform a reaction over and over and over and over again. Think about how much energy it would take the cell if it had to perform thousands of reactions in a second, and it needed to make a new enzyme for each one of those reactions. Instead, it can make just a handful of them and they can perform the reaction over and over and over and over again. Our bodies and everything in nature, our cells, are very lazy. They want to take the path that requires the least amount of energy input if possible. So they are not changed and they are not used up by the reactions that they are catalyzing. Now, enzymes can be modified a little bit. We don't just want to have these enzymes in our body, and the only way that we can regulate their behavior is by either not making them or completely destroying them. Remember, these are very energetically costly to make for the cell, so we don't want to be breaking them apart and remaking them if we can help it. We'd much rather just add something to them and kind of block their action for a little while, or remove something from them that allows them to function. That way we can say, okay, you can stop for a while, but I'm going to let you go back to work in a few minutes. We call those activators and inhibitors. So activators are chemicals that bind to secondary sites on the enzyme and improve the function of the enzyme. So things often like vitamins or ions like electrolytes, these will find somewhere other than the active site to bind, and it slightly changes the shape of the enzyme because it's chemicals interacting, and this kind of turns the enzyme on. It wakes it up so that it can now actually fit the substrate, whereas before maybe it couldn't fit the substrate, or it was very difficult for it to fit the substrate properly. Now it has the most optimal shape to take in that substrate. There are also inhibitors, molecules that stop or inhibit or reduce an enzyme's ability to function. And there's two types that we're going to talk about. The first one is competitive inhibition. This is where an inhibitor actually fits in the active site of the enzyme itself. So if this inhibitor is in the active site, the actual substrate cannot fit in there and be broken down or be catalyzed. So competitive inhibition, they are competing with the substrate for the active site. Non-competitive inhibition is when the inhibitor binds to somewhere else on the enzyme, not the active site. So in this case here, the active site is this V shape on the enzyme, and this other site is this half moon shape. We call that an allosteric site. Basically just means the other place. And when these allosteric inhibitors, these non-competitive inhibitors, when they bind to that site, it causes a shape change in the enzyme, just like any kind of chemical reaction would. But in this case, it very specifically changes the shape of the active site, so it can no longer fit the shape of the substrate. And therefore, the substrate can't bond, the reaction cannot occur. So competitive, they compete over the active site. The non-competitive inhibitors, they don't want to compete. They're going to find the allosteric site and cause the shape change so that the active site is no longer the proper structure. And that is it. We're going to go into how these enzymes can be sped up and how they work, or slowed down, what other factors affect their functions, and things like that when we meet in class. So let me know if you have any questions.